Welcome, my digital pathology trailblazers. If you are here, okay, I see people joining, and you should be able to see me and hear me. Let me know if you do, and let me know where you're tuning in from. And of course, you know which platform I'm struggling with, Instagram. But I think I'm there on Instagram. I think so. If you are on Instagram, I don't know how you can let me know, because I don't think I can see the Instagram comments. But if you are anywhere else where you can comment, let me know, and let me know where you're tuning in from. I'm in Pennsylvania, 6 a.m. in the morning, and freaking cold this week. Let me tell you how many degrees. I don't know, many, like in Celsius. Yesterday it was -14. Some other day it was like recently, 2 days ago, it was -20. So in Fahrenheit, that's like, I don't know, 3. Very cold, very cold. So let me know where you're tuning in from. And if you hear me well, I'm gonna send a comment. And then okay, I see people joining. So if you are joining, let me know where you're tuning in from so that I can see that you guys hearing me well. My Polish coffee cup. Good morning. I have, okay, more people joining. Fantastic. I'm going to give you a few updates before we gather, and then we're going to jump into the paper. But what was recently, so if you have not been to Path Visions, then, okay, and we have people from the UK. Fantastic. Welcome. If you have not been to Path Visions, that was November 2024, then you can get the recordings if you would want to learn what happened there. Also, if you want to check out the vibe of the conference, there's going to be a few vlogs and few videos coming out on my channel. So if you are on my list, which I assume you are because you're here on the DigiPath Digest, number 21, by the way, we are almost... No, if it was in years, we would be legal to drink. And I don't know if tobacco is also 21 in the US. But anyway, number 21 today. Welcome, everybody. And when you're joining, let me know where you're tuning in from. But to be respectful of those who joined on time, let us dive into the papers. So the first paper today is... Oh, come on. Is it working? Yes. Can PSMA PET detect intratumor heterogeneity in histological PSMA expression of primary prostate cancer? And then there were two tracers, gallium radioactive PSMA gallium and fluoride radioactive. So these are tracers that you put into the body as like a contrast tracer, something that's supposed to attach and then be easily imageable, if that's a word, easy to image. And this is a group from Freiburg, Germany. So what did I do here? The PSMA is prostate-specific membrane antigen. This PET technology, positron emission tomography, is a promising candidate for non-invasive characterization of prostate cancer, PCA. And this study evaluated whether those tracers, PET tracers with tracers, and they used these two tracers, is capable to depict intratumor heterogeneity of histological PSMA expression. Why is this important? Because those cancers that express a lot of it, they react better to treatment. So, of course, they were looking for a non-invasive way to image this, because pathology, even if it's slide free, or unless maybe you can do it something like really intraoperatively, but it's still surgery is invasive, right? So what did they do? 35 patients with biopsy-proven primary prostate cancer, no evidence of metastatic disease, or any prior interventions. They were enrolled into this trial, this study. And then they did this PSMA PET, they did computer tomography, and they did either this one marker, this gallium, for 20 patients, or the fluoride for 15 patients. And then they did the radical prostatectomy. So they had the whole prostate, and then they could do all the IHC they wanted. And so they also, in addition to that, I'm removing my markings because then I cannot do more markings. They did ex vivo CT, and then histopathological sections were prepared. And then they manually defined these areas with different morphologies, and they defined an H score of IHC PSMA. What is an H score? It's a score where you say, okay, how much of this tumor is affected with how strong of a expression of a marker. I think it can be up to 300 is the maximum score. Anyway, there is a way of calculating this. This is a classical way of evaluating immunohistochemistry, how much of the marker is there. And this was calculated with assistance of AI, which I'm very grateful for, because then you get much more accurate results when you do that, rather than estimating the H score and all these other numeric score that you're actually visually estimating and not really like counting those cells, even though the score is based on the number of cells or percentage of cells. And I see more people joining. Thank you so much. Whenever you come in, then let me know where you're tuning in from. And if you are listening to the recording of this, because I can imagine that not everybody, especially in North America, wants to wake up at 6 a.m. and in the West, it's even earlier. Let me know also in the comments on whichever platform you're viewing it, where you're coming in from, where you're tuning in from. So they used AI for the H score, and they had these prostate cancer areas with similar H score, and then they unified in segmentation on ex vivo CT. So they checked, because with CT you can do the CT sections, and they checked, okay, how does it match with the IHC of the real sections? And they calculated different metrics, right? One of them being standardized uptake value, SUV. I love it. So the SUV and different other metrics were calculated. They checked the agreement of the co-registered tumor areas to gross tumor volume in PSMA PET as well. And what were the results? The results here. Oh, and we have people from Dublin. Welcome, Dublin. Amazing to have you. So great to see you. And I know some of the people joining. Some of them are digital pathology trailblazers who have been that way for a long time. So super happy to see you. So results. For the histological prostate cancer areas, IHC PSMA expression correlated significantly to the SUV, standardized uptake value, mean and max. And there was an approximately linear correlation between the H score and SUV mean value in tumor areas larger than 400 micrometers in histology. And tumor areas with strong PSMA expression show larger overlap to this gross tumor volume in PSMA PET after co-registration than tumor areas with very low PSMA expression. And the tracers, those two tracers, there was no difference between the two tracers. And the conclusion is that those tracers, the PSMA PET with both tracers, gallium and fluoride, is able to detect changes in histological PSMA expression with prostate cancer lesion, allowing biologically targeted radiotherapy. Yes, but does it then? They don't say that you don't need to do IHC anymore. And it's telling me that my microphone is not having so much battery. I hope we can last till the end. And if you're just tuning in, let me know where you're tuning in from. We have a lot of papers today, so I'm just gonna keep going. Okay, HistoCol AI, call because it's collaborative. HistoCol AI, this group from Valencia, Spain, and also some members from Quebec, Canada. I used to live in Quebec, not in the city. Uh-oh, my mic is disconnected. Give me 1 second. I need to find my other mic. Can you still hear me? Let me know if you can still hear me, because maybe it's good enough to have my mic from the camera. If you could do that. In the meantime, I'm gonna try to find my other mic, which I should have ready. I have everything ready for vlogging at conferences. Okay, you guys can still hear me well? Then I'm just not gonna bother. If you stop hearing me at any point, just give me a comment immediately and I'll activate my other fluffy microphone. It's super fluffy today because of the cold. Okay, let me know what temperature it is where you are to make me feel bad that it's warmer or maybe colder. People from Quebec, Canada should have it colder. Okay, anyway, so this HistoCol AI is an open source web platform for collaborative digital histology image annotation with AI-driven predictive integration. Sounds super cool. And why does it sound super cool for me? Because doing annotations and providing pathology input for image analysis projects is notoriously challenging because there's not really any infrastructure that can be used. Sorry, I'm trying to direct this camera better to me. Should be good. Anyway, so it's notoriously challenging, and it's in the image analysis world, even in institutions where this is something you do every day. It's basically a string of workarounds how to give pathologists who not always do the image analysis access to those images, to review the markups, to provide annotations for the image analysis scientists. There's I have seen very few good workflows. So that's why I'm excited about this HistoCol AI, and it's open source. And we said the people from Valencia did this and from Quebec. So they are very optimistic because they say digital pathology is now a standard component of the pathology workflow. That would be so cool if it was standard. Not everywhere, but in this group it is. And we're getting there. We're getting there. And every single one of you is helping with it because you guys are here and wanting to learn about it. And I see more and more people joining. So let me know where you're tuning in from. So what happened there? Okay, this digital pathology is supposed to be standard. But there's a significant challenge in developing computer-aided diagnostic systems for pathology because there's lack of intuitive open source web applications for data annotations. And I couldn't agree more. And also there's another problem with data annotations. They disappear after the project. Although you have some companies that basically offer this as a service, and I believe that they keep their annotations to reuse them, but most of the time, no interoperability between annotations, between different softwares, and basically hundreds or thousands of work of pathologists and tissue specialists who are annotating for model development is just disappeared because of the lack of interoperability. So this paper proposes a web service, and the next step for me will be to check out this web service that effectively provides a tool to visualize and annotate digitized histology images. And it's mostly, well, it can use different formats, but they have it working best on TIFF format. And this integration, they say, not only revolutionizes accessibility, but also democratizes the utilization of complex deep learning models for pathologists unfamiliar with such tools. Okay. I don't know how I would need to read the paper, because they're talking about providing annotations and collaborating on annotations and not deploying models, but maybe they do deploy models. So they present a use case centered on the diagnosis of spindle cell skin neoplasm involving multiple annotators. So that's what's going to be described in this paper. And they also conduct a usability study showing the feasibility of the developed tool. Interesting. And this is open source. So my problem as non-computer scientist for these open source things is when I go to GitHub, I never know what to do with this application. QuPath, at least I can download. Well, maybe it's easy, so I'll check. And you can check as well and let me know in an email or in the comment of this video, because that would be promising. You know what my video editor suggested that I do? That I do a virtual annotation sessions. So if you're interested in joining me for a live stream of annotations, like, you know, the gamers, they just play the game and you watch them play the game, and they're in the corner, and then they have their gaming screen. So something like that, but for annotations. So that would require a tool for annotations and also the images, right? So if you guys want to take part in something like that, let me know in the comments. Just write annotations, and I will know that you're interested in that and that you would like to join. And you can write also the organ that you would like to have annotated. If you have a specific question right now and that you're struggling with for annotations, let me know in the chat. Annotations and the organ that you want to have annotated, and I'll figure out how to organize a live stream annotating session. Going back to our papers, the next one is interesting, very interesting. It's called Enhancing Its Radiology, European Journal of Radiology Open, and it's enhancing detection. Okay, perfect. We have already some annotation live stream request for colon cancer. Okay, and if you guys have data sets and you would just want me to access the data sets and just like annotate with you live, if that's obviously allowed for the data set that you have, let me know and we can figure it out. That would be so cool. I think that would be super cool. And then you can just have the link and always know how to annotate. Okay, so this is a group from Egypt, and they did this study, the enhancing detection of previously missed non-palpable breast carcinomas through AI. And my main question when I was reading it was this question: what about the patients? But I'll tell you why in a second. So, to investigate the impact of AI reading digital mammograms and increasing the chance to detect the missed breast cancer, they did a study. They have AI flagged early morphology indicators overlooked by the radiologist, and then they correlated them with missed cancer pathology types. So mammograms done between 2020 and 2023, they had a lot of breast present those mammograms, over almost 2,000 mammograms. And they were analyzed in concordance with the prior 1 year's results, 2019, 2022. Assumed negative or benign were analyzed in concordance with the prior one. Yeah. So The results here were that the prior mammogram with AI marking compromised. Sorry for reading things twice. 54%, so 555. And in the present mammograms, AI targeted 904, 88% carcinomas. The descriptor proportion of asymmetry was the common presentation of missed breast cancer carcinomas, 64%, in the prior mammograms, and the highest detection rate for AI was presented by distortion and then followed by grouped microcalcification. So these are radiologic features of malignancies that AI was able to detect, and they predicted malignancy in previously assigned negative or benign mammograms, showed sensitivity of 73, 89, specificity of 89 and accuracy of 78. In conclusion, here is reading mammograms with AI significantly enhances the detection of early cancerous changes in dense breast tissues, and AI detection rate does not correlate with specific pathology types of breast cancer, highlighting its broad utility. So the AI just detects cancer and not a specific type. So basically it flags, and then you can do whatever diagnostic workup is necessary later. And subtle mammographic changes not corroborated by ultrasound but marked by AI warrant further evaluation. So if AI marks something, you try to evaluate it. And my question before is, what about the patients? So they used this AI, this was a retrospective study. So my question was like, okay, so if they detected AI with AI, they suspect cancer, it was detected with AI, what did they do with those people? Because this is recent, right? 2020 and 2023, 2019 and 2022. Like, what did they do? Did they like go to the people and said, hey, well, actually, without AI, it was missed. You have to come back. In the paper, they didn't do it. It was retrospective, and there wasn't any. They didn't contact the patients or anything. And I want to trust that. So no, let's put it that way. It's one of the ethical questions that comes up when using AI, and feel free to comment on this. And this is abstract, so we would need to dive deeper in the paper, but that's going to be something that the people who deal with AI ethics and what are the boards, the not ethical boards, there is like a special board that allows people to do studies. I don't remember the name. Put the name in the chat if you remember. It's not ethical board. It's some commission. Anyway, that's going to be a question that you're going to be asking, like, okay, and also in terms of, is it then, like, if you don't have this tool available, or if you didn't use the tool, are you then responsible for missing something if you didn't really have the tools necessary to visualize it? I don't have the answer, but definitely something that comes up with all those AI enhanced workflows that are not evenly distributed across the world. And that's why ethics is a separate, and bioethics is a separate discipline in life sciences. But that was basically something that came to my mind when I was reviewing this retrospective study. So if you have any thoughts or comments, let me know in the chat. And in the meantime, let's go to the next paper. Advanced tissue technologies of blood-brain barrier organoids as high-throughput toxicity readouts in drug development. I like it because I'm a toxicologic pathologist, so but I'm not gonna dwell too long on it, but I'm going to tell you what it is. So this was Roche Pharma research. And shout out to Roche, because Roche just became a Digital Pathology Place sponsor. So whenever my sponsors publish, I do want to give them a shout out. And this is a group from Basel, Switzerland. And what did they do? They did those organoids. So the recent advancements in engineering complex in vitro models, CIVMs. Oh my goodness. By the end of the abstract, I don't remember the abbreviations anymore. But this one we will remember, because it's just BBB, such as blood-brain barrier, BBB organoids. So they did these BBB organoids, and they offer a promising platform for preclinical drug testing. However, their applications in drug development, and especially for regulatory purposes of toxicity assessment, requires robust and reproducible techniques. So here they have an adapted set of orthogonal image-based tissue methods, including H&E, IHC, immunohistochemistry, multiplex IF, and also even MALDI, which actually that's the only instance where an abbreviation is easier than the full name, which is matrix assisted laser desorption ionization mass spectrometry imaging, mass spec, to validate those complex in vitro models for drug toxicity assessment. So just a step back. What are these orthogonal methods? So basically you take different methods and you show with different methods the same thing. So that's something that's gonna show for the regulators that an approved method, or a method that is already accepted or already cleared, or something that there is already out there widely accepted, shows the same thing that the novel thing shows. So they had a bunch of them, right? H&E, IHC, and multiplex IF, and even MALDI, and they used AI. Thank you so much for doing this, because when I hear multiple markers evaluated visually, I cringe, because it's difficult, or if not impossible. And they use this to increase the throughput, first of all, and probably increase the reproducibility and the reliability of histomorphologic evaluation of apoptosis for in vitro toxicity studies. So apoptosis was the feature that they used AI for, and they quantified apoptosis with AI. And their data highlight the potential to integrate advanced morphology-based readouts, such as histological techniques and digital pathology algorithms, for the use on complex in vitro models as part of standard preclinical drug development assessment. And why is this important? There is a rule of 3 R's or 4 R's, basically for toxicologic studies, and a big part of those studies happens on animals. So you want to refine your study design, replace the animals, and something else. Let me look up 3 R's for you on my phone. What is the 3R rule in toxicologic studies?
The 3R rule in toxicological studies stands for replacement, reduction, and refinement.
Okay, this replacement, reduction, refinement, basically meaning as little of those animals as possible to still have a scientifically valid conclusion. So these complex in vitro models are part of this initiative, or can be part of this initiative, because the more information you can get from in vitro, the more reduction, refinement, and replacement you can do in the next studies, which are the animal studies, right? So super excited about that. Cool method. And now let's start. Okay, and I have some comments that making an annotation session could be fun, and different organs you would like to just hang out for annotations. Perfect. I'll figure out how to do it. I'll figure out what tools. And if you are a vendor and would like to sponsor such an annotation session in your tool, I'm totally open to that. Let's talk about that, and let's do some annotations together. There are cool commercially available tools to do that, but the point of the session is going to be, okay, showing you the structures to annotate, and if you have something for specific projects, let's do it. And now let's talk about implementing trust in non-small cell lung cancer diagnosis with a conformalized uncertainty-aware AI framework in whole slide images. When I read this title, I'm like, what is it? Implementing trust. But basically, it's about implementing trust in those AI models with different computational metrics that, I'm a pathologist, but I'll try to explain as best as I can from the abstract. So, ensuring that trustworthiness is fundamental to the development of AI. I would say development, I don't think for the development, but definitely for deployment, that is considered societally responsible. Yes, particularly in cancer diagnosis. And here we go into the ethics field as well. Okay, how trustworthy are those algorithms, right? So, because misdiagnosis can have dire consequence, of course, but that can happen with or without AI. And current digital pathology AI models lack systematic solution to address trustworthiness concerns. Yes, I totally agree with that. And there is not like one framework how to validate those. Now the awareness of having to do that is a lot bigger than maybe in like 2012, 13, 14, when I was starting to work with that. Then you would basically publish data on your training set, would be your test set, and you would just like have a super overtrained model or image analysis solution and publish that and say, oh, that's fantastic. And people who did not know the math, the statistics behind it, didn't even know it was a flaw of the study, right? I was only pointed to that by my computer science colleagues telling me, hey, tell your pathology colleagues to stop validating on your training set. And I'm like, what? And then I understood. Anyway, so the trustworthiness, there are some concerns. They are arising from model limitations and data discrepancies between model deployment and development environments. This is super important. Let me see if I can like change my color. Let's do discrepancies. Where is it? Discrepancies, deployment and development. So let me talk a little bit about that. So we develop, so the if you want to build a medical device, let's say you want to build an AI model that is a medical device everybody can use. Do you have data from everybody to train on? No. So you figure out ways to make this algorithm generalizable. But is there a guarantee that it's so generalizable that it's going to work on everything? No. Thomas, agreement in statistics is so much fun. I know, questionable. It's like another branch of science that when I dive deep into it, I like think I understand it for like a week when I am deep, and then I forget it. But basically that's something that needs to be taken care of. So, but development and deployment, right? Even if you have like the most generalizable model on the planet, if there's data from outside your cohort, there is no guarantee it's gonna be the same type of data, right? And you trained an algorithm or model on a certain type of data. So what can we do to make sure that your algorithm is even an appropriate tool for the new data that's coming in? And this is what those researchers did. They developed that TrueCam, a framework designed to ensure both data and model trustworthiness in non-small cell lung cancer subtyping with whole slide images. Non-small cell lung cancer was basically their use case. You can probably do it for anything. And they had this TrueCam, and in this TrueCam, there are different computational things that they are doing. One of them is the spectral normalized neural Gaussian process for identifying out-of-scope inputs. And this is something like, would you think this is controversial in the image analysis world? It is controversial because there are like two different camps. Sorry, I'm not making my paper big. Apologies for that. Let me know in the comments. Let me know when you don't hear me, when you don't see stuff. I see the comments. So every time there is something that I can improve, let me know. So the controversy here is, okay, you have a data set. Is consistency or is like reliability using one algorithm for this data, for like your whole data cohort, and this is consistent? Or is it consistent or like reliable to use the appropriate method for different parts of your data? And I definitely am the proponent of the other method, where you need to use the tool that is appropriate for your data. And it was a bigger issue before deep learning, because deep learning is definitely more generalizable than the hard coding or thresholding, or what do you call it? The handcrafted feature approach. Before we had deep learning, where, for example, for IHC detection, you would define the size of the object, the threshold of your of the DAB, of the brown stain, and basically you would manually say, okay, most of my data looks like this. This is gonna be applied. But you will always have data that are kind of out of scope, they call it. I don't think you can call it out of distribution. That's something else. But out of scope, like let's say a different run of IHC from the same lab that was a little fainter, right? What do? Do you run the same algorithm on it, and then you have a readout from this algorithm that does not really match the tissue? Or do you exclude this from your data pool, and then you have different methods on your data cohort? I am a proponent of different methods that give you a good result that matches the tissue. There are people who think differently, but to address this, our TrueCam was developed. So right, so the first thing was, okay, we check which of our data are out of scope, and this is fantastic because you can already say, okay, I will use this particular tool on part of my data. This maybe has to go with a different algorithm, or maybe it has to be visually assessed by the pathologist. Then, on the second thing, ambiguity guided elimination of tiles to filter out highly ambiguous regions, addressing the data trustworthiness. I think it's good. Like, if it's ambiguous, why do you force a tool that you don't know if it's gonna work? And then conformal prediction to ensure controlled error rates. And then they systematically evaluated this framework across multiple large-scale cancer data sets. And I think they used our friend of TCGA, maybe, leveraging tissue cancer genome atlas, leveraging both task-specific and foundation models. So they did it for different models, right? The narrow ones and the foundation, to illustrate that AI model wrapped with TrueCam significantly outperforms models that lack such guidance. So you can wrap your model in TrueCam, basically, to flag the stuff that you already can predict that your model is not reliable on that. I would like to have this discussion with a computer scientist, because that seems to me like a cool way of triaging the data that comes into your pipeline into, okay, something has to go to a pathologist, the rest can be done with AI plus pathologist or something like that. Let me know what you think about it in the comments, especially my image analysis folks here on the line. So then, what did we say? AI model wrapped in TrueCam significantly outperforms models that lack such guidance in terms of classification accuracy, robustness, interpretability, and data efficiency, while also achieving improvements in fairness. These findings highlight TrueCam as a versatile wrapper framework for digital pathology models with diverse architecture designs, promoting their responsible and effective applications in real-world settings. Cool. So you can basically wrap whatever you develop into this, and you will know that what you developed is going to work well on a certain tiles, certain data types, image types. Cool. I think it's cool. And I see more people joining a little bit later. Let me know where you're tuning in from. And we are gonna move on to the next paper that talks about QuPath, which is open source. Cool. There's a video on the channel how to annotate in QuPath. And it's funny also, every now and then I make those videos because it's super useful tool, but then I don't use the tool, and before the next video I have to reteach myself how to annotate in QuPath. So, but now I can watch my own videos to teach myself how to annotate in QuPath. And also there's one for Aperia image scope. We have people from Chile. So cool. Hello to Chile. What time is it right now? It's not that much of a time difference between us. No, maybe it is. We'll see. We'll see. Let me know. Let me know. Okay, so QuPath extension for glomerulosclerosis and glomerulonephritis characterization based on deep learning. So deep learning plugins or additions to QuPath are a recent invention. This is a group from Germany and also from Spain and Italy. Why did I not highlight them? Please. Sorry for that. And what happened here? The digitalization of slides has opened new opportunities for pathology, such as application of AI. Let's see, where was it published? In what? Computational Structure Biotechnology Journal? Something like that. Compute Struct Biotechnology. And I didn't check the impact factors for you. Okay, it's 8:41 a.m. in Chile. Okay, good. Good time for this kind of educational activity to start the day, or maybe not start, maybe, depending on what time people wake up. So, but specialized software is necessary to visualize and analyze these images. That is true. You will not open them in like Windows Image Viewer. One of these applications is QuPath, bioimage analysis tool. And other than the videos, there's also a podcast with QuPath founder or person who started, Pete Bankhead. And they propose, this study proposes GNCNN, the first open source QuPath extension specifically designed for nephropathology. It integrates deep learning models to provide nephropathologists with accessible automatic detector and classifier of glomeruli. And glomeruli are the basic filtering units of the kidneys. And I'm reading this and I'm like, okay, how is it useful to the nephropathologist? Depends, right? So like, if you want to show glomeruli to a nephropathologist, that is not that useful. They know where the glomeruli are. But if you want to quantify or like count the ones that are sclerotic and count some other ones like glomerulosclerosis and glomerulonephritis, then it's useful because you don't make them count stuff or guesstimate. You show them the markups and they say yes, no, useful or not useful. But basically, when it goes into quantification, that is useful. At least that's what I think as a pathologist, because I know where glomeruli are in the kidney. We could annotate glomeruli. Maybe we can get access to some kidney data sets and annotate in QuPath or in this other thing that we just discussed, the open, what was it? Call AI? Call AI? HistoCol AI. Where is it? HistoCol AI. Ha! I remembered. Okay, QuPath. Let's focus. Let's focus. And I'm talking to myself. You guys probably are more focused than me. I'm jumping. Okay, so the glomeruli, basic filtering units of the kidneys, such as glomerulosclerosis, glomerulonephritis, and this GNCNN offers a user-friendly interface that enables nephropathologists to detect glomeruli with high accuracy and categorize them as either sclerotic or non-sclerotic. Okay, that could be useful. Achieving a balanced accuracy of 98.46. Furthermore, it facilitates the classification of non-sclerotic into 12 commonly diagnosed types of glomerulonephritis. No kidding, that's like super useful. I didn't know that there are 12 types. But I'm also not a nephropathologist, so excuse
my lack of knowledge in this area. Commonly diagnosed types of glomerulonephritis with top 3 balanced accuracy of 84. And this GNCNN provides real-time updates of results, which are available at both the glomerulus and slide level. Who is this? Maybe we can contact them and show it to us on the channel. Checking if I know anybody. No, but we can contact them. Cool. I'd like to see it live. I'd like to see it in the working. So if you know anybody of the authors, send them this live stream. And maybe they're going to be interested in showing it beyond the publication to the digital pathology trailblazers. And if I have the bandwidth, I might reach out to them. So this allows users to complete a typical analysis task without leaving the main application of QuPath. So here I want to emphasize something. Without leaving the main application. And I think we talked about it a little bit at some point, that, and I called it like a first world problem. Oh, a pathologist needs to click another window to access some results. But if you multiply it by the amount of slides that the pathologist needs to look at every day, and by like the amount of movements with the wrist that they need to do to go click somewhere else, it is a hurdle. It is a digital workflow hurdle. So here they said you don't need to do that, which is fantastic. I like it. And this tool is the first to integrate the wrong highlighter. First to integrate the entire workflow for the assessment of glomerulonephritis directly into the neuropathologist's workspace, accelerating and supporting their diagnosis. I would want to learn about their workflow and workspace. How do they integrate it? Maybe I can invite them to the podcast? We'll see. We'll see. Let me know if you want that. And then another thing of annotations, a lot of annotations. And don't worry, I know we're, how many more? We have 2 more, so bear with me. It was a long list. Sometimes we only get like 5 and 3 of them are not relevant, but today we have a lot of relevant ones. So annotation-free deep learning algorithm trained on H&E images predicts epithelial to mesenchymal transition, phenotype, and endocrine response in estrogen receptor positive breast cancer. Super cool. Let's start with epithelial to mesenchymal transition. What is that even? So you have 2 main types of tissue. Well, I think you have 3. No, but 2 main, that is epithelial and mesenchymal. So epithelial is basically everything that's on surface from the inside and from the outside. And mesenchymal are muscles, blood, and basically connective tissue. And tumors, they start as epithelial and then can transition to mesenchymal. So they basically change their tissue type because tumors are basically cells out of control, and they do that. And it correlates with the endocrine response. The epithelial to mesenchymal transition is something visible. You can see it in the slide. So I'm excited about these type of applications because if there is something you can see and then you can detect and quantify that actually corresponds or predicts treatment, molecular expression of something, or like something that you cannot see, that's fantastic because then you can detect it in the image and you have an image biomarker, right? So this is a group from China, somewhere else, no, China, London, UK. And let's talk about it. So recent evidence indicates that this endocrine resistance in estrogen receptor positive breast cancer is closely correlated with phenotypic characteristics of epithelial to mesenchymal transition, EMT. EMT, is that not? It's emergency medical technician. Well, not here. It's epithelial to mesenchymal transition. Love the abbreviations. You know that. Nonetheless, identifying tumor tissue with the mesenchymal phenotypes remains challenging in clinical practice. So they validated the correlation between the transition, epithelial to mesenchymal transition status, and resistance to endocrine therapy in ER positive, estrogen receptor positive breast cancer. And again, I don't have my paper large, sorry for that. From a transcriptomic perspective, to confirm the presence of morphological discrepancies in tumor tissue of ER plus breast cancer classified as epithelial and mesenchymal phenotypes, according to the epithelial and mesenchymal transition related transcriptional features, they trained a deep learning algorithm, and this is the architecture of efficient net v2 architecture to assign the phenotypic status for each patient utilizing H&E slides. Oh, here they use the Cancer Genome Atlas database, TCGA. And the classifiers more accurately identified the precise phenotypic status, achieving an area under the curve of 0.886 at the tile level, and the AUC was 0.9 at the slide level. Furthermore, we evaluated the efficacy of the classifier in predicting endocrine response. So first they checked, okay, with image analysis, they detected, is it epithelial or mesenchymal? And then they checked, does it actually correlate to this response or to this resistance in response to this endocrine therapy? And the classifier achieved a predicting accuracy of 81.25% and 88.7%. Slides labeled as endocrine resistant were predicted as the mesenchymal phenotype, while 75.6 slides were labeled as sensitive, and they were epithelial phenotype. So again, mesenchymal are the ones resisted. You can see mesenchymal in the image. Epithelial, or at least to the 75%, are the ones that are sensitive, and you can see that they are epithelial. But sometimes it's difficult to visually assess. So if you can have an AI assistant, that's great. And so their work introduces an H&E based framework comparable of accurately depicting as epithelial to mesenchymal transition phenotype and endocrine response to ER positive breast cancer demonstrating potential for clinical application and benefit. I love these applications where you have something visually identified and then you can relate it to something actionable in the patient care journey. Okay, my friends, my digital pathology trailblazers, if you joined late, let me know where you're tuning in from, because I see a lot more people than we had at the beginning. And we have one more paper today that is titled Leveraging Explainable AI. So that ties into what I just said. If you see it, it kind of gives you a level of explainability, at least to a pathologist, because it's a feature of the tissue that is associated with if some specific pathophysiology, right? And they already know, but now if you can quantify, that's even better. So leveraging explainable AI and large-scale data sets for comprehensive classification of renal histologic types. Okay, so this is a group from Korea, and this is Science Reports Korea. Anything else? I think it's just Korea. So, what did they do? They also used AI in genitourinary pathology. And the recent research focused on classification of renal cell carcinoma subtypes. Well, the most recent research that we just reviewed in QuPath focused on classifying glomeruli, but also, just joking, a classification renal cell carcinoma is a big thing. In general, like classifying all the different types of tumors is a big thing in this image analysis community and in this digital pathology / AI space. And that's where the focus is still and was for a long time, cancer, right? Not inflammatory disease, not so much well, some specific metabolic diseases like liver steatosis. We discussed this one digipath just ago or two. Anyway, big focus on cancer, right? And the broader categorization into non-neoplastic normal tissue, benign tumor, and malignant tumor remains understudied. This gap in research can be attributed to limited availability of extensive data sets, including benign tumor and normal tissue, in addition to specific type of renal cell carcinoma. So basically, if you only had tissue from renal carcinoma, that's what your model would be focusing about, right? If they obviously have more different types of renal tissue, and they have a model that answers a different question that classifies or figures out different things. Their research introduces a model aimed at classifying renal tissue into three primary categories: normal, non-neoplastic, benign tumor, and malignant tumor. And I'm reading this and I'm like, okay, this here, differentiating between these two, benign and malignant, is going to be tough. Between normal and tumor should be okay. And that's already very useful if you want to, for example, deploy some specific model just for tumor. You will not deploy it in the normal tissue, right? Utilizing digital pathology host slide images from nephrectomy specimen. Need to visualize nephrectomy specimen, 2,535 patients. That's a lot. That's a huge data set compared to majority of papers. From multiple institutions, the model provides a foundational approach for distinguishing these key tissue types. The study utilizes a set of over 12,000 whole slide images, and it's going to be 1,300 normal tissue, 700 benign tumors, 10,000 malignant tumors. So we already see that we have more or less comparable normal and benign, and we have a lot of malignant tumors. So they already have a bias in their data. They have tumors, a lot of tumors. Employing the ResNet 18 architecture and multiple instance learning approach, the model demonstrated high accuracy with F1 scores of 0.934 for normal tissue, and we see benign tumors is lower, 0.684 for benign tumors, and we're back to higher, 0.878 for malignant tumors, right? The overall performance was also notable, achieving a weighted average F1 score of 0.879 and a weighted average area under the receiver operating curve of 0.969. So let's talk about it. What do they call the overall performance? Implying, let me just find the exact wording of this. Yeah, the overall performance. So like you put all those categories, and if most of them are good, it's going to be high. But clearly this not normal, the benign tumors are underperforming. So if it was me, I would lump all the tumors into tumor and then give it to a pathologist. And because my comment to this one was, cool, but what for? I don't know. What is this department? Is this medical center? I'm checking if it's more like computational, biomedical engineering, health science technology, data. What there is a hospital pathology. Anyway, sometimes it's useful to just develop a technology, and then the use case for the technology shows up later, or this can be leveraged in a different context. But I also, full disclosure, I didn't go deep into the paper. This is my opinion based on the abstract. Let me know your opinion. Let me know also if you have any questions that are related to this live stream, unrelated to this live stream, but related to digital pathology. We're focusing on digital pathology because whenever there are enough questions to start Q&A live streams, I'll start them. If you're interested in a live annotation live stream, let me know in the comments as well. Write annotations and add the organ that you're interested in, regardless whether you're here live or whether you're watching the recording. And if you think this thing is useful, this DigiPath Digest is useful, give it a thumbs up, subscribe to the channel. That helps it get to more people that have similar interests to you, and helps us promote digital pathology. I think this was the longest live stream ever because it's almost an hour. In 5 seconds, it's going to be 1 hour. So let's call it for today. Thank you so much for joining. Thank you so much for staying till the end. Like, subscribe, and see you next Friday. Bye. Take care. Stay warm if your place is cold.